Assalamualaikum and hi. In this video, we Group 3 from Kuliah 2 will be presenting about DNA extraction experiment. This group consists of 4 members which are Alia, me myself, Fatimi Rushaida, Mardiatul Mariam, and Nurin Akila. So the learning outcomes for this experiment is to observe the extracted DNA from the onion and to identify the role of chemicals in the process of extracting DNA. The next part of this video will be explained by Aida.
Let's move on to the introduction. As we know, bodies are made out of units called cells. When you cut yourself, you damage the living cells, and when the cut heals, new cells grow and make more skin. Every cell in our body has a master control region called the nucleus. And as we know, inside the nucleus there is a material called deoxyribonucleic acid, and common name is DNA. DNA is a molecule that is present in the cells of living organisms. It contains the genetic information found in every living cell and is the hereditary blueprint of life. In this experiment, we use onion to run the experiment. Why? Because onion has a low starch content and which allows the DNA to be seen clearly.
All the materials used in this experiment are household items. These are the procedures. Next, we will look at a video recorded by Nurin during the experiment.
Nathan and Hal are another tomorrow. For the discussion of this experiment, it involved the breaking salts to release the DNA at the first place. The chopped onion was mixed with distilled water and blended together into an onion solution. After that, the onion solution was mixed with the detergent solution, since the detergent causes the cell membrane to break down. The solution then placed into the hot bath water not more than 15 minutes. It is because after 15 minutes, the DNA would begin to break down. Then, the solution was placed into ice water bath for 5 minutes to stabilize the DNA. After that, the solution was filtered for 2 times, which were using the muslin cloth for the first time, and after that using the filter paper. The filtered mixture mixed with ice-cold alcohol using the medicine dropper. The purpose of mixing the filtered mixture with alcohol was to precipitate the DNA because the DNA is not soluble in alcohol. After a few minutes without disturbing it, the stringent molecules were observed between the mixture and the alcohol. And the stringent molecules were the precipitation of DNA. The precipitation of DNA was collected using the correct way to spool the DNA, which was using a constant motion and pressure slowly. There are a few precaution steps in this experiment. Firstly, during the mixing of filtered mixture with alcohol, make sure that the alcohol was slowly added into the filtered mixture by closing the end of the dropper to the side of test tube. This is to make sure that the DNA would not be destroyed and could not be used. Secondly, make sure the onion solution and the detergent in the Ziploc were sealed tightly before putting into the hot water bath to prevent the mixture from being spilled. And lastly, make sure the mixture was cooled in the ice water bath before the filtration. It is because the cool water temperature will stabilize the DNA. So I think that's all from me. Thank you.
Hello, it's me Aida again. So now I will be presenting about the questions that we got from the lab manual. So the first question is, state the function of each chemical or components below in DNA extraction experiment. So we have salt, detergent, and ice-cold alcohol. So let's see the answer. For salt, we use salt to neutralize the DNA charge and make the molecule less hydrophilic, which means it becomes less soluble in water. We use detergent because detergent contains a compound called sodium lauryl sulfate that removes fats and proteins. It will also cut through cell walls because the membranes surrounding the cells are made out of fats and proteins. The detergent pulls apart the membranes and releasing the DNA. Last, we use ice-cold alcohol to increase the yield of DNA. Let's move on to the next question. The question is, why is the onion mixture processed through the blender? The blending of onion is the mechanical operation that increases the total onion surface area, which means there are more onion cells with surface exposure. Some onion cells will burst from the chopping process. This part is to break down cell walls to let the DNA and other things out of the cell into the solute. The more cells that are on the surface, the more that break apart, and the more of the original DNA that we can collect. The last question is, why do you need to cool the mixture in ice water bath? DNases or restriction enzymes that destroy DNA are present in the cell cytoplasm. They are there to protect the cell from invasion by viruses. Once the nuclear membrane is destroyed by detergent, the DNA is now susceptible to the DNases and will quickly be degraded. However, these enzymes are temperature sensitive and cooling the solution slows down the process of degradation. I think that's all from me. Thank you.
So from this experiment, we can conclude that DNA extracted from the onion can be observed as white stringy mass, and that salt makes the DNA become less soluble in water, detergent dissolves the cell membrane of the onion, and ice-cold alcohol is used to increase the yield of DNA. These are our references, and these are the credits that specify the task done by each member. So I'd like to thank all the members for their efforts, and also Thank you for listening. That will be all from us.